So today we're going to be talking about oxidative phosphorylation, which is really our second part of cellular respiration. Last class we talked about how we take one molecule of glucose and we systematically break the bonds in that molecule to create 6 carbon dioxide molecules, as well as, remember, those electron carriers, NADH and FADH2. And I told you to think of those as Uber drivers because they are going to carry electrons, and they're specifically going to carry electrons to a portion of the mitochondria that will perform oxidative phosphorylation. So in our notes we've drawn this reference cartoon. Remember that we are in the mitochondria, that is that double membrane organelle, and specifically we are going to zoom in and focus just on structures that are located in the inner membrane. Remember, the inner membrane is that highly folded structure that forms cristae, and embedded in that membrane are both the electron transport chain and ATP synthase. And we're going to see that the electron transport chain works to, as its name suggests, transport electrons, and that's going to allow this system to build and create a proton gradient, and then that proton gradient is going to be used by ATP synthase to perform a process called chemiosmosis, and chemiosmosis allows us to make ATP. And this is where the bulk of ATP is going to come from in cellular respiration. So let's look at how this process works. So we are going to start first with just the oxidative portion of oxidative phosphorylation. And so we're just going to look at the electron transport chain for now. In your notes, I've given you the cartoon blank version from your textbook of the electron transport chain. And your job as we go through this is just to label the components and take notes on what's happening at each step. So we're going to start here, and we're starting specifically at protein 1 of the electron transport chain. And at this portion of the electron transport chain, NADH, one of our Uber drivers, is going to come in and finally deliver its passenger. It's finally going to let go of the electron it's been carrying, and it's going to donate that to protein 1. Notice that NADH is losing an electron, and so it has now become oxidized, and it's reverted back to that NAD+ form. So oxidation is lost. Remember, oil rig. So we've oxidized our electron carrier, and now our electron can move, and we can follow it through the yellow arrow to protein 1. This donation of electrons is a downhill reaction. So because it's downhill, a little bit of energy is getting released. And that's really important because protein 1 is actually a proton pump. And so what it's going to do, remember, pumps pump things against their concentration gradient. And so it's going to use the little bit of energy that's released from this electron to pump protons, those hydrogen ions, from an area of low concentration to high concentration. Because ultimately we're going to see we want to build a high concentration of protons in this intermembrane space. All right, so electrons been donated, NADH has been oxidized, the energy release can be used to drive this pump. Now our electron can move to the second molecule, which is that Q molecule. Again, Q stands for ubiquinone. It's not that important that you remember the long name, but just remember this is a molecule that sits inside the lipid bilayer of the inner membrane. And it's going to pick up an electron not only from protein 1, but also down here from protein 2. Remember, protein 2 is our weird little protein that is actually a peripheral protein that hangs out on the matrix side. And notice that our second type of Uber driver is also present at protein 2. Protein 2 is going to interact with FADH2. FADH2 will also release its electron. It's going to drop off its passenger, and it becomes oxidized back to FAD. And so now Q has picked up electrons from both protein 1 and protein 2. And these electrons are going to continue their journey to protein 3 here. Protein 3, again, is also a proton pump. And so it's going to steal that energy that's been released from this downhill movement of electrons to, again, pump protons against its concentration gradient. So now we're adding to the amount of protons that are on the outside of this membrane. The electron, as we follow the yellow arrow, continues to our next molecule, which is cytochrome C, or Site C. And Site C really kind of just works as a hopping place so that these electrons can then move on to protein 4. And protein 4 is really the end of the line for the electron transport chain. Protein 4 is also a proton pump, and so again, it's going to use the energy from that downhill transport of electrons to drive protons against the concentration gradient. And so now we have created a very, very high concentration of protons on the outside in the intermembrane space, and we have a low concentration of protons on the inside or the matrix side of our membrane. Okay, but we still have kind of an issue here because the electron that goes here needs to leave protein 4. It can't just stay at protein 4 forever. And so we actually have to have a final electron acceptor, and that is where oxygen comes into play. I've been telling you that we, in order to perform cellular respiration, and even when we drew our little simple chemical reaction, I told you that you have to have oxygen. This is why. Oxygen is going to function as the final electron acceptor in this process. In doing so, it is actually going to use the energy that's donated by that electron, and it's going to interact with a proton, and it's going to perform a chemical reaction that allows it to build water. So this is why I told you when you breathe out, your breath is kind of wet. It has that moist quality to it. That's because your mitochondria are making water, because oxygen is accepting that final electron and building water, and now we have to get rid of that water vapor. So oxygen here functions as the final electron acceptor so that this process can continue over and over and over again. Our Uber drivers, NAD and FADH2, can continually bring in our electrons. Oxygen, as long as it's present, can always be here to pick up those electrons and get rid of them, and we form water in doing so. And so we are oxidizing our electron carriers in order to create a high concentration of protons, which are going to be used in the next step of oxidative phosphorylation. So now we can finally move to the last portion of our cartoon, and we can focus on what ATP synthase actually does. And again, it performs the phosphorylation portion of oxidative phosphorylation. So if we zoom out and we now add ATP synthase to our diagram, there is an ATP synthase protein after every single electron transport chain. And again, because that inner membrane of the mitochondria is highly folded, there are thousands of electron transport chains shoved into one mitochondria. So we have a huge possibility of making a lot of ATP after every single electron transport chain. So what ATP synthase does is it performs a process called chemiosmosis. Chemiosmosis just refers to the fact that we have a gradient of a chemical. Remember, we have a high concentration of protons here, a low concentration of protons here. So we're going to use that chemical gradient to drive the creation. Specifically, we're going to drive phosphorylation to make ATP. So what happens is, remember, we have this high concentration of protons, and in an ideal environment, these protons want to move down their concentration gradient. They want to diffuse from areas of high concentration to low concentration. What's really nice about ATP synthase is it provides a channel for those ions to do so. So within this protein is actually a proton channel, and these protons can start to diffuse down their gradient. They can move from areas of high to low concentration. And again, because this is a passive process, this is a downhill reaction. And so every time a proton moves through ATP synthase, they are releasing a little bit of energy. And that's great because the bottom portion of ATP synthase is actually responsible for taking ADP, remember adenosine triphosphate, plus a free phosphate, and using that released energy, it's going to build ATP. So it's going to take ADP plus phosphate, it's going to combine those together, and we have now made an ATP molecule. And again, we've made some of that energy that our cells have to have in order to survive. And so by taking advantage of a chemical gradient, by having that chemical move down its gradient, we can steal that energy essentially and use it to build a very important molecule. And so now, if we go back to our piggy bank where we were keeping track of how much ATP cellular respiration makes, we can add that for every electron transport chain that provides this, that creates this proton gradient, that allows ATP synthase to perform chemiosmosis. This process will make between 26 to 28 ATP molecules. So again, one glucose molecule makes enough NADH and FADH2, reduces enough electrons so that we can create this many molecules. And now if we add that to our total, we can make around 30 to 32 ATP per one molecule of glucose. Remember last time we just started with one glucose molecule, and from one we can make 32 ATP. Pretty cool. A lot of ATP can be made in this process. So that is oxidative phosphorylation. We are using oxidation. We are oxidizing our original electron carriers. That oxidation allows us to build a proton gradient, and that proton gradient can be used to phosphorylate or add a phosphate to ADP so that we can create adenosine triphosphate.